Hello, so today I'm just going to revise my science and technology notes, and this is for biotechnology. So let us begin. Immunity is basic sense that body's ability to guard itself. Okay, now here comes the important thing. There are you just need to remember this WBCs, RBCs, and platelets. Then I want to emphasize here that this is the new type of therapy that is next Cas9 therapy, and it is related to beta cell blood cancer. So what happens is in normal cancer we do chemotherapy, and in chemotherapy our normal cells and our cancer cells both get destroyed together. So this is a major issue that we face in chemotherapy treatments that are currently a viable option. So now in this immersion technology that we have is CAR T cell therapy. So what it does is patients own T cells attack the cancer cells. Okay, so how great it would be if the patient's own cells attack the T cells. And here we have the meaning of this CAR T is chimeric antigen receptor. Okay, just remember this, and it can be directly asked that what are the applications of following CAR T technology. So this is CAR T technology. Then we have international HapMap project. This is just mapping of genetic variations in human species, not a big deal. Then we have gene drive technology, creating genetic changes in insects to prevent them from spreading diseases. Okay, altering Mendelian inheritance. Now here comes the important part, that is RNA silencing. Before proceeding to RNA silencing, let me first tell you this, that this is the basics. We have DNA, we have mRNA, and then we have protein. Okay, so what happens is there is this process. This mRNA is called messenger RNA. This messenger RNA just copies the information from DNA to itself, and when it copies the information from DNA to mRNA, this is called as transcription. In this transcription, this is a recipe, like a recipe book for creating a protein. It is like a recipe book for creating a protein. So this is called transcription. And then from mRNA we get translation. Translation is just the decoded or decrypted information that is needed for a cell to generate a protein, generate a type of protein. So we have transcription, then we have translation, and the main thing between this is mRNA. So now similarly our cell generates protein with the help of DNA. Likewise a virus cell or any bacteria also generates the same, or it replicates using this similar methodology. So what we are doing is we are silencing RNA. This RNA is very important thing in the cell, in cell functioning. Okay, so now in RNA silencing, what we are doing is done by body itself to chop off viral RNA before it can replicate. Whenever we are affected with cold, fever, or anything, suppose this is a virus, and this is its DNA strand. It tries to multiply and divide itself so as to increase its population in our cell. It enters through our wound or whatever, it enters through our lungs when we respire some toxic air or something like that. Okay, so what it does is tries to replicate itself. So by RNA silencing, what we are doing is we are just cutting like a scissor, how we cut a paper. Likewise in the same manner we are just cutting this RNA. So what happens if we cut this RNA is that it does not replicate, it does not do this process called translation and transcription, it cuts itself. So this is RNA silencing technology. It is very helpful in this tobacco plant, where the nematode bacteria infects the tobacco plant. Okay, you can read about it in detail. I am here just to revise the concepts. How to silence this RNA. We add a nematode specific gene into the plant. Okay, this is just things. Then we have DNA, mRNA, protein, mRNA, antisense. Okay, you just need to remember the basics of mRNA, and that is it. Don't go into the intricate details. UPSC won't emphasize much upon it. Mitochondrial diseases are inherited from mother only. This is a PYQ. This is a PYQ of, I guess, 2024 or 2025, if I'm not wrong. Direct hit. That mitochondria is known as powerhouse of the cell. This powerhouse is directly inherited from the mother. Just remember that. Father has no role in this. Then okay, pharmacogenomics studies how a person's genes affect the response to drugs. Okay, this is a term in use, just remember that, pharmacogenomics. Then we have jumping genes. This is also very important. Jumping genes, Jumping genes are DNA sequences that can move from one location to another within the genome. And one example is line 1. So UPSC can ask in one word association that what is line 1. Line 1, it can tell you about astronomical unit and something like that. We just need to remember this is one word association. Then we have National Biofoundry Network. Here we have Bio E3 policy. In Bio E3 policy, the government aims to use biotechnology for environment, then for economy and for employment. Just remember that. And we have hydrogel is also a PYQ, I think from 2023 or 2024, I think 2024. Hydrogel contact lenses are much in use near infrared for firefighters. They are used in medical field. Okay, this was the question asked, which of the following hydrogels are used. I think this was in 2024. Then we have DNA. In DNA we have, okay, DNA I have explained the basic things here. We have cells, then inside cells we have chromosomes, inside chromosomes we have string of DNA, a segment of DNA, and then we have gene. Okay, then I will go to gene editing. In gene editing, what do we know? We have these following methods. CRISPR-Cas9 everybody knows, and it was very much in news. Similar to CRISPR-Cas9, we have following methods: talents, and then we have ZFNs. Then we have base editing and prime editing. Base editing and prime editing are also very much important. You need to know the difference between these two. What is base editing? What is prime editing? I think I have included this difference also. But let me see where I have included the difference. Yeah, here we have the difference, direct difference: base editing and prime editing. Base editing, change in one letter, and prime editing is rewrite a small DNA sequence. Okay, this is more complex. Prime editing is more complex. This is just base level editing. That is why we have the name called base editing. Here you can see complexity simple of base editing. Then we have use case 5-point mutation. Here we have in prime editing we have more complex, and then fixed complex mutations. Then we have DNA editing is permanent, RNA editing. Yes, you need to remember this. DNA editing is always permanent, and RNA editing is just temporary process. Then we have then we have TREGs, that is known as regulatory T cells. I have told you in the just a moment ago that T cells are very much important for our body to fight against any virus or any bacteria causing harm. Specialized T lymphocyte cells. Okay, suppress immune system. Main function is to suppress immune system, maintain self tolerance, prevent body from attacking its own tissues. Okay, and key gene is FOXP3 gene. Okay, the Nobel Prize was also dedicated. See here, peripheral immune system. So what happens is, suppose you take example of vitiligo. Otherwise, okay, let's take vitiligo. So if a person has a vitiligo, then what happens is its own body cells attack its own skin cells. Okay, attacks its own skin cells, and what happens is, due to the attack of skin cells, the melanocytes get destroyed, and eventually the skin turns white. Or pale. That means your skin does not produce any color. Why does this happen? Because your normal body cells attack their own cells, and they mistake this normal cell, and they think that this is the real enemy. They think that this is a virus, this is a foreign body. And this exact gene was identified, and this gene is called as FOXP3 gene, and this will be a revolutionary technology, and this itself is very much important for a prelims examination. Then we need to remember that do not enhance immune response, do not attack pathogens directly, they suppress, regulate immunity. Okay, then we have microRNA, small non-coding RNA, acts after transcription. Okay, we have seen this, binds to mRNA, blocks protein synthesis, or causes mRNA degradation. Then we have it reduces or blocks protein formation. Then we have TREGs is suppress immune system. This is just a summary. FOXP3 prevent autoimmunity, mRNA. Then what do we have? This is done for biotechnology. Okay, I have just covered it in a hurry, in a very brief. If you want this PDF, you just comment. I will share the link of the Telegram channel, and then you can download this PDF. This is for biotechnology. These are scripts, notes that you just need to go through. You don't need to master, you don't need to have a mastery in all of the terms that are mentioned here. You just need to go through the basics, like the FOXP3 gene. This is very much in use. Then you need to go through this microRNA, then base editing, prime editing. Then we have CRISPR-Cas9. This you just need to go through this. Then we have another important thing, if I have to emphasize, is Bio-E3 policy, very much in news. Jumping gene, also very much in news. Then we have pharmacogenomics. Then we have RNA silencing. Yeah, that is it. So we have just handful of topics that you need to go through. You don't need to read entire PT 365. So yeah, that is it for the video. Thank you.